A warm welcome to dear students. The present lecture has been prepared in accordance with the UGC curriculum of BSc first year botany students. In view of a large number of students who fail to understand the lower plants or botany, the present lecture deals with the diversity of microbes. Microbes or microorganisms are very small living things which cannot be seen with the aid of naked eye. They are typically uni- and multicellular microscopic organisms widely distributed in air, water, soil, sea matter, and with the bodies of plants and animals. They are included in the group of lower plants. Microbes constitute a part of our world and are present in both natural and man-made environments. Microbes have diverse and special qualities which enable them to survive in such unique places such as air, soil, oceans, rivers and streams, icebergs, hot springs, the human intestines, within and outside the plant pots, and even in oil wells. Therefore, microbes are omnipresent with a high degree of adaptability as they spread over the entire biosphere, that is lithosphere, the hydrosphere and the atmosphere. This group of organisms show a great diversity in their cell structure and functions. Some of the major groups of microorganisms are prions, second viroids, third is virions or viruses, fourth rickettsia, fifth mycoplasmas, sixth bacteria. Now let us discuss them one by one. Prions, they are small proteinaceous infectious particles that lack nucleic acid. Stanley Prusner in 1982, an American neurologist, identified them for the first time while diagnosing a neurological disease in sheep called scrapie. Prions are disease-causing agents, and a major portion of infectious agent is a glycoprotein called PRP or prion protein. Hence, Prusner coined the term prion for proteinaceous infectious particles. 9 animal diseases now fall in this category, including the mad cow disease that emerged in cattle in Great Britain in 1987. All the diseases of this group are called spongiform encephalopathies because large vacuoles develop in the brain of the hosts. The human diseases caused by prions are Creutzfeldt-Jakob disease, commonly abbreviated as CJD, Gerstmann-Straussler-Schindler syndrome, and fatal familial insomnia. These diseases are caused by conversion of a normal host glycoprotein called PRPC for cellular protein into an infectious form called PRPSC for scrapie protein. Now second group, that is viroids. Theo Diener in 1971 discovered new infectious agents which were smaller than viruses and named these subviral pathogens as viroids. A viroid consists of very short strand of RNA, about 300 to 400 nucleotides, without any envelope or a capsid. The best example of viroid is potato spindle tuber viroid. Current research on viroids have revealed that they resembled with introns for not coding any polypeptide. Moreover, the viroids have been identified as the plant pathogens only. Chrysanthemum stent, Chrysanthemum chlorotic mottle, and cucumber pale fruit or some other diseases of plants caused by viroids. For their replications, viroids depend completely upon host machinery, including the necessary enzymes. The next group of microorganisms, that is viruses. Viruses, as we know, are non-cellular, submicroscopic, obligate parasites and nucleoproteinaceous in nature. They were reported for the first time by Carolus Clusius from tulip flowers, and later a German botanist, Adolf Meyer, in 1868 described a mosaic disease of tobacco, now commonly called as tobacco mosaic, which was later found to be caused by a virus known as tobacco mosaic virus. Viruses were purified in crystalline form by Stanley in 1935, and complete viron was studied by F.C. Bowden. The size of viruses varies from 20 to 350 nanometers. On the basis of nucleic acids, viruses are of two types: RNA viruses and DNA viruses. Plant viruses usually contain RNA as their genetic material, and animal viruses possess DNA as their genetic material. However, there are some exceptions. For example, human immunodeficiency virus or HIV, being an animal virus, possesses RNA as its genetic material, while as cauliflower mosaic virus, which is a plant virus, contains DNA as its genetic material. Viruses vary in their shape also. Some viruses are helical, for example, tobacco mosaic virus or TMV, rabies, mumps, etc. Some are polyhedral, for example, adenovirus and poliovirus, while others are complex in their structure, for example, bacteriophages. Viruses are conveniently classified into many categories, such as the bacterial viruses or bacteriophages, the blue-green algae viruses or cyanophages, the plant viruses, the animal viruses, and mycoplasma viruses. Bacteriophages are virulent or avirulent. Virulent phages cause lysis of the host cell after infection, while as later remain latent and are referred as temperate phages and replicate along with the host chromosome. Bacteriophages of T group are the examples of virulent phages, and phage lambda is the best example of temperate phage. Viruses of blue-green algae resemble with those of bacteriophages of T group. Some of the plant viruses are tobacco mosaic virus, potato leaf roll virus, tomato bushy stent virus, tomato leaf curl virus, etc. Some of the animal viruses are adenovirus, influenza virus, poliovirus, rabies virus, vaccinia virus, etc. Sixth report of International Committee for Taxonomy of Viruses or ICTV records a universal taxonomy scheme of viruses comprising one order, 71 families, 9 subfamilies, and 3,600 virus species. Hundreds of viruses still need to be added to the scheme because of unavailability of data. Viruses do not reproduce like those of other organisms, rather they replicate by their sole nucleic acids, either DNA or RNA within the host cell. Viruses of different groups show some common mechanism of replication, such as adsorption and penetration, biosynthesis, maturity, and release. Next group of microorganisms is the mycoplasmas. Mycoplasmas are smallest known aerobic prokaryotes without cell wall. Pasteur was the first who discovered them while studying the causative agent of pleuropneumonia, and hence mycoplasma are also called as PPLO or pleuropneumonia-like organisms. The first successful cultivation of a mycoplasma, the bovine pleuropneumonia agent, was first reported by Knockaert and Rox in 1889, and after the isolation of L variants by Killenberger in 1935, mycoplasmas were given a separate taxonomic status and were detached from viruses with which they were earlier grouped. Mycoplasma lack cell wall, hence they are irregular and variable in shape. The cells may be coccoid, granular, pear-shaped, cluster-like or ring-like or filamentous. The cells are small and range from 0.3 to 0.9 microns in diameter. The cells are covered with a unit lipoprotein membrane. The cytoplasm contains ribosomes, nucleoplasm-like structure with DNA and RNA. There are three genera of mycoplasmas. One is the mycoplasma, second a coliplasma, third is the thermoplasma. Three types of reproduction has been seen in mycoplasma. They are: number 1, binary fission in equal or unequal parts; second, by budding of small units; third, by the breaking up of protoplasm of a single cell into numerous small units called elementary bodies. The elementary bodies are the smallest known cells so far. Mycoplasma were not considered to be the causal agents of plant diseases. However, it was in 1896 when Hugo de Varies reported some 27 plant species of his garden infected by mycoplasmas, but it was Doy and his co-workers who reported that mulberry dwarf and ester yellows, which were suspected to be virus diseases, were in fact caused by mycoplasmas. Mycoplasma diseases of human or primary atypical pneumonia, commonly abbreviated as PAP, found in mouth, pharynx and genitourinary tract. It is caused by Mycoplasma pneumoniae, repeated or habitual reproduction failure in women, and birth of underweight children is due to the infection of Uriplasma urinalyticum, and infertility in man is caused by Mycoplasma hominis and Mycoplasma fermentatus. Another group of microorganisms which we talk about is Rickettsias. It was Fracastorius in 1546 who first described an epidemic which was later identified as typhus fever. The disease has played a large role in history ever since people began to congregate densely under conditions where body lice flourished. The microorganisms causing this group of disease were named Rickettsias to honor H.T. Ricketts, who discovered the agent of Rocky Mountain spotted fever early in 1900 and later died of typhus fever while investigating its etiology. Rickettsias are the smallest prokaryotes, 0.3 to 0.7 microns wide and 1 to 2 microns long. They are larger than any of the viruses. They vary in their shape. They may be cigarette-shaped or rod-shaped, spherical or ovoid, and often pleomorphic. Rickettsia multiply by transverse binary fission. They are intercellular obligate parasites and easily cultivated in live chick embryos. They are primarily parasites of insects and secondarily in man and other animals. The Rickettsia are responsible for a number of diseases known as the spotted fever group. These include epidemic typhus caused by Rickettsia powaziki and transmitted by lice, endemic murine typhus caused by Rickettsia typhi and transmitted by rat fleas, and Rocky Mountain spotted fever caused by Rickettsia rickettsiae and transmitted by ticks. In humans, rickettsial infections damage the permeability of blood capillaries, which result in characteristic spotted rash. Another group of microorganisms is the bacteria. Antony von Leeuwenhoek in 1675 has seen bacteria for the first time with his crude microscope, which could hardly magnify things up to 150 times. Bacteria are distributed worldwide except in the pits of volcanoes, deep strata of rocks, or blood of normal animals. They are present under the icebergs as well as in the hot springs. Some species tolerate a pH lower than 1 and some tolerate a pH higher than 13. Thiobacillus thiooxydanus is the best known acidophilic bacterium. On the basis of nutritional methods, bacteria are distinguished into three main groups, that is autotrophs, chemotrophs, and heterotrophs. Autotrophs directly use sun's energy and with the help of bacteriochlorophyll, which is then converted into chemical bond energy of sugar and other carbon compounds. A few examples of autotrophic bacteria are Rhodomicrobium, Ectothiorhodospira, etc. Chemotrophs, on the basis of inorganic oxidation, sources are divided into iron bacteria, sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria, methane bacteria, and carbon bacteria. Heterotrophs obtain their energy by decomposing organic material absorbed from the surroundings. The decomposition is carried by the secretion of extracellular enzymes secreted by the bacteria. Heterotrophs are further subdivided into saprophytes and parasites. Bacteria have been divided into 10 orders as per the Burgess Manual of Determinative Bacteriology. They are: number 1, Pseudomonadiales, Chlamydobacteriales, Hyphomicrobiales, Eubacteriales, Actinomycetales, Caryophoneales, Bigetoales, Myxobacteriales, Mycoplasmatales, and Spirochaetales. On the basis of their morphology, bacteria are divided into many forms. For example, bacillus or rod-shaped bacteria, they are further subdivided into diplobacillus, palisade bacillus, and streptobacillus. Coccus or spherical bacteria, they are subdivided into monococci, diplococci, tetracocci, streptococci, and staphylococci, and spiral shaped bacteria, for example spirillum and vibrio. On the basis of their staining, bacteria are classified as gram-positive and gram-negative bacteria. On the basis of the flagella on their body, bacteria are classified as atrichous, monotrichous, amphitrichous, lophotrichous, and peritrichus. Bacteria are basically unicellular, but sometimes they form loose irregular mosses or incipient filamentous. The average size of a bacterial cell is about 2 microns, though the range lies between 0.2 to 40 microns. Bacteria generally reproduce by binary fission, as true sexual reproduction is absent. However, gene transfer occurs in bacteria through many processes such as conjugation, transformation, and transduction. With this, we conclude this lecture, dear students. Thereby, we see a great diversity in the microbial world. Thank you.